So this beta oxidation, we define it by saying that is the process by which the fatty acids in the form of acyl-CoAs, are broken down in the mitochondria. We know what the mitochondria is, or in peroxisms. We also know what peroxisms are, such as organelles in the cell, to generate acetyl-CoA. And we know that acetyl-CoA is the first molecule which gets into the Krebs cycle, or the TCA or the CCA. We know that the first step usually you have to condense acetyl-CoA and oxaloacetate, if you remember what Krebs cycle does. So, in other words, what you're saying, that oxidation or beta oxidation of fatty acids ultimately will give us what? Acetyl-CoA. But the fatty acids have to first be activated by adding a component called coenzyme A, and that's why they're usually referred to as acyl-CoA. So acyl-CoA molecules, so they are broken down, of course, in the mitochondria and peroxisome. So in other words, if somebody was to ask you which organelles where is beta oxidation occurring, it's either mainly in the mitochondria and partly in the peroxisome of the cell. And of course, this occurs majorly in cells of the liver, the kidney, and the heart. Yeah, we know that the heart is fairly surrounded with quite some lipids to a healthy extent, and then maybe sometimes to an unhealthy extent, the kidney as well as the liver. And you've seen that the liver is the horse or the donkey of the body, because most of the metabolic activities are occurring. Now, fatty acid oxidation doesn't occur in the brain. Anybody to answer why would it not occur in the brain? Admit this guy. Why would this process not occur in the brain? Maybe in physiology you've touched about it. Anyone? Why would it not occur in the brain? Victor?
So in the
Victor.
brain, the only has utilizes carbohydrates because of the blood-brain barrier that prevents
Yes.
other biomolecules from entering the brain.
Yeah, you've raised two key points there, which I wanted us to know, take note of. First of all, I think we already know, and I've mentioned this previously, that the brain exclusively uses glucose as its source of energy. Number two, of course, physiologically, you know that the brain has the blood-brain barrier. So, of course, it prevents fatty acids from getting into the brain, and therefore, it's highly unlikely that beta oxidation is going to happen in the brain, correct? Now, so typically, this would be what a fatty acid looks like. For instance, palmitic acid. Palmitic acid is C16. It has 16 carbons with a carboxylic acid. So forget about this S-CoA first with a carboxylic acid. So if this should be an oxygen, a carboxylic acid here. So all the carbons should be technically 16. That is palmitic acid. Now, when this palmitic acid is activated, in other words, acetyl-CoA, so not acetyl-CoA, but coenzyme A is added on the palmitic acid. Now it's now being referred to as palmitoyl-CoA because it's now an activated fatty acid, what we are referring to as an acyl-CoA. So this is an acyl-CoA. It's just an addition of coenzyme A on this, and that is activation. Once it's activated, then it can now be broken down into acetyl-CoA, and that is what we are going to look at, how that then happens. So for beta oxidation to occur, there are about 3 steps that are followed. There are about 3 steps or 3 stages that are followed for activation of, for beta oxidation to occur. Now, the first step is activation of the fatty acids in the cytoplasm. You've already seen what is happening in activation. First of all, this would not have the acetyl-CoA. Once the it doesn't have the coenzyme A, but when the coenzyme A is added, then it's activated. So that is what the first step is, activation of fatty acids. And this is happening in the cytoplasm. That's the first stage. Then the second stage is that the activated fatty acids get into the mitochondria. And the transportation into the mitochondria is via what we call the carnitine shuttle. The carnitine shuttle, I'll get to it in a moment. We'll be able to understand how that happens and how it's a bit unique. And lastly, we have the actual oxidation now, the actual oxidation of the activated fatty acid, the last step. And the actual activation of the fatty acids, the last step, steps in it. So oxidation properly by itself has 4 steps. So first of all, they're activated by adding a component of coenzyme A, so it forms an acyl-CoA. Then it's transported into the mitochondria. Remember, the activation is happening in the cytoplasm. So, and we know that this process of beta oxidation is happening in the mitochondria or in the peroxisome. So, the activated fatty acid needs to move to the mitochondria or the peroxisomes, and the movement specifically in the mitochondria is done by enzymes that are referred to as the carnitine shuttle. Then, of course, beta oxidation. Now, let's go through each and every step and see. So, first of all, we have the activation. So, this activation is done by an enzyme called the fatty acid thiokinase, or it's also known as the acyl-CoA synthetase. The acyl-CoA synthetase. So we're going to generate an acyl-CoA, and of course, this is happening in the cytoplasm. Now, this thiokinase, or the fatty acid thiokinase, or the acyl-CoA synthetase, requires ATP. It requires coenzyme A. Coenzyme A is usually abbreviated like this, and magnesium ions. That is what is needed. So then, after this, you're going to generate a fatty acyl-CoA and water as the products. Okay? So you have a fatty acid in the cytoplasm together with coenzyme A, and this process requires energy in form of ATP. Then you're getting a fatty acyl-CoA. This is a fatty acid attached to coenzyme A. Of course, water is the other byproduct, but you're also going to utilize 2 phosphates so that then you generate an AMP in the process. That is the activation. So in activation, you're just generating a fatty acyl-CoA by simply adding coenzyme A to the fatty acid. Okay? Of course, on the carbon attached to the carbon that is attached to the carboxylic group. Now, the second step is what we are calling the transportation into the mitochondria. And the transportation is done via the carnitine shuttle. The carnitine shuttle is what is doing the transportation. Remember, in the cytoplasm, here outside in the cytoplasm, we've generated an activated acyl-CoA, fatty acid that is attached to coenzyme A. So we have an enzyme called acyltransferase 1. We generally refer to these enzymes as CAT. Of course, it's carnitine acyltransferase, so CAT. So we have CAT1 and CAT2. CAT1 is outside, CAT2 is inside the cell. So CAT1 is outside the cell in the cytoplasm. CAT2 is in the matrix of the mitochondria. Now, if you remember, when we were talking about enzymes and enzyme activities generally, we classified enzymes into how many groups? If anybody would remind us? Tulisema enzymes ziko grouped into how many groups? Okay, so we have 7 classes of enzymes. Who can remind us the 7th class ilikuwa inaitwa nini? The last group of enzymes ilikuwa inaitwa? Precious?
Translocase.
Translocases, okay. Yeah, so this last class of enzymes we used to refer to them, or we refer to them as translocases. Now, translocases or teams, we have team and Tom. Is it team and Tom? Yeah. So these translocases are specifically found in the matrix of the mitochondria, either in the outer membrane or the inner membrane. So these carnitine transferases, they are translocases. These enzymes are actually translocases. So either found in the outer membrane or the inner membrane. So translocase of the outer membrane, that will be referred to as the TOM, or translocase of the inner membrane will be referred to as the T. So actually, CAT1 is a TOM, CAT2 is a TIM. So this CAT or carnitine, what does it do then? You have an activated fatty acid like that. So what happens is that the carnitine removes the coenzyme A bit and it becomes attached to the fatty acid. So in other words, it's separating the fatty acid from coenzyme A. Okay? When that happens, then these molecules are able to be transported through the membrane into the matrix of the mitochondria. Now, once this happens, so carnitine is going to carry both. So carnitine, first of all, is attached to the fatty acid. Then we have the coenzyme A that will be able to move together with the carnitine attached to the fatty acid into the. So once they are in the matrix, you are having now coenzyme A separate and carnitine separate. Then CAT2 will remove the carnitine from the fatty acid and attach the coenzyme A onto the fatty acid, so that now you still have the activated fatty acid like that. And then, of course, it shuttles back the carnitine to the outer membrane. So that is what we are referring to as the carnitine shuttle. We know that shuttling is movement between 2 places, okay? So frequent movement between 2 places. So the carnitine enzymes are shuttling between the outer into the side, the outer membrane and the inner membrane of the mitochondria. And that's why they're referred to as the translocases. So they're shuttling in between here. And as they shuttle in between, they are moving along with the molecules. So they separate coenzyme A from the activated fatty acid. So together with the coenzyme A, there's movement into the matrix. And once they're in the matrix, then the CAT2 will remove the carnitine from fatty acid, join the coenzyme A to the fatty acid, so you remain with your activated fatty acid. So in general, it's how carnitine looks like as a molecule. But I didn't really want us to focus on the structures per se, because we already dealt with structures previously, and we can't exhaust all structures. So of course, I'm sure we never talked about the structure of carnitine, but it's just for you to appreciate that it would be looking like this. So what then the beta oxidation can start, because now you have fatty acid getting into the matrix of the mitochondria. So it's at the point of which activation can occur. think this just still the same elaboration. This is just the same elaboration. You have a fatty acid, it's activated with acyl carnitine, it gets into the mitochondria. Then from the mitochondria, that is where beta oxidation will start happening. And beta oxidation, of course, will generate acetyl-CoA, and acetyl-CoA will get into the TCA cycle, but we also have reducing equivalents that will get into the electron transport chain. So we know that Krebs cycle is going to give us energy. The electron transport chain is also going to give us energy, especially when you're utilizing reducing equivalents such as NADH and FADH. Okay, so this is just an elaboration, or rather a bigger elaboration of what is happening, including this process. Now let's see. So at step two, what is happening? What I've just explained is that the acyl groups from the acyl CoA is transferred to carnitine to form acyl carnitine. Okay, and this is activated by carnitine acyl transferase. This is happening in the outer mitochondrial membrane. So then we are having the acyl carnitine is shuttled into the mitochondria by translocase. Okay, then the acyl group is then transferred back to the CoA in the matrix by carnitine acyl transferase 2. So then finally carnitine is returned into the cytosolic side of the by the translocase in exchange for an incoming acyl carnitine. Okay, that is exactly what we've been talking about here. Okay, so yeah, translocases, they're moving this, the carnitine translocase, of course, is moving this into the matrix. Then, of course, there's joining of carnitine back to the acyl CoA. So that is exactly what I've been explaining. Now, the third step, and we said the third step has 4 steps. Now, in the third step, this is the actual oxidation. This is the actual beta oxidation of the fatty acid. So we have the first step is oxidation, and we are using FADH. Okay. The second step is hydration. There's an enzyme hydratase there. The third step is another oxidation and using NADH. And the fourth step is a splitting step or thiolytic cleavage by using an enzyme called thiolase. So these are the 4 steps. Now, let's just stick to the example of this particular molecule, that is palmitic acid. And this palmitic acid has 16 carbons, like I've already said. And now it's been activated, and now it's an acyl CoA, like this. Now it's palmitoyl CoA. That is the molecule. So we see how is it now going to undergo beta oxidation. Now remember the first carbon here that is usually linked to the carboxylic acid, it's like that. Then the second carbon is referred to as the alpha carbon. The next one is the beta carbon. So let's just see what is happening. So it's important for us to know that this first carbon is not being referred to as the alpha carbon. Instead, it's part of the carboxylic acid. So the alpha carbon is the next carbon from the carbon that is forming part of the carboxylic acid. And of course, now it's forming part of the acyl CoA. So we know that the first reaction is oxidation of the acyl CoA. And this is done by acyl CoA dehydrogenase. Now, what is happening is that another bond is being introduced between the alpha and the beta carbons. Okay, another bond is being introduced between the alpha and the beta, so that you are creating an unsaturated acyl CoA, or what we'll refer to as an enoyl CoA. And for that, you require FAD as the hydrogen acceptor. You require FAD as the hydrogen acceptor. And of course, the enzyme here will be acyl-CoA dehydrogenase. We'll have the enzyme as that is the alpha and the beta. So we want to introduce a double bond between these two so that now we create an enol-CoA. Now, in this case, of course, the hydrogen that is being removed at this particular point is going to be accepted by FAD so that then it forms FADH2. So in this case, you see what is happening now. The CoA bit is remaining, but now a double bond has been introduced between the alpha carbon and the beta carbon. Okay, of course, there's loss of hydrogen there. Now we have a molecule here we are calling an enol-CoA or alpha beta unsaturated acyl-CoA. We also refer to it as trans 2-enol-CoA has been generated. Remember, the chain is continuing, so we say that this is a 16, but it's just dealing with the first two carbons. So it is dealing with the first two carbons, but these first two carbons, we're only seeing that a double bond is being introduced between the second and the third carbon, which we are referring to as the alpha carbon and the beta carbon. That is where hydrogen bond is being introduced. And remember, we are forming a trans molecule here. This is a trans. That's why we are referring to it as trans 2-enol-CoA. It is trans. We know the difference between trans and cis configuration. So we're generating a trans configuration like this. So this is a trans enol-CoA that is being generated. So that's the first step, oxidation. Now the second step is hydration. The second step is hydration. So the hydration is occurring at the double bond to the beta hydroxyacyl CoA, or rather, where the enol CoA is being removed. And this double bond is being removed by addition of oxygen, or rather, a component of water. So we are calling it hydration, but the component of water is being added here. So this double bond will go. And that is activated by an enzyme called enol-CoA hydratase. Enol-CoA hydratase. So you're introducing an O here at the beta carbon. So you have an OH. And now since the double bond went, of course, we'll have two hydrogens. Initially, you have one hydrogen, but you now still have the two hydrogens here. And remember, it's still the trans. It's still trans. Still trans. On the trans is where the oxygen is being introduced. And on the other trans is where the hydrogen is being introduced. So you have an H2 here. So this particular molecule in pink, we're calling it L orientation beta hydroxyacyl CoA, because we introduce hydroxyl group this particular point on the beta carbon. So we're calling it the beta hydroxyacyl CoA. So that is the second step, the hydration step. Now the third step, you remember, is another oxidation, because we said you do oxidation, hydration, oxidation, and then cleavage or thiolysis. We're breaking it down. So in this oxidation, the beta hydroxyacyl CoA will produce a ketoacyl CoA. And the moment you see a keto, it means a double bond being introduced with oxygen, thermal. So the OH will go, and instead a double bond bound to oxygen will be introduced. And that requires, of course, NAD as the hydrogen acceptor. And the enzyme here is beta hydroxyacyl dehydrogenase. Okay, so this initially had OH, now it has CO, and of course now this becomes a keto. Okay, it is moving from an aldehyde to a keto. It becomes a keto here. So now we are referring to this molecule as ketoacyl, but it is still occurring at the beta carbon. So it's the beta carbon, rather the third carbon, that is undergoing lots and lots of transformation. So that is the third step. Now the final step is cleavage. The final step, you want now to break the molecule at this particular point. The fourth reaction is cleavage of the two carbon fragment by splitting the bond between the alpha and beta carbons. And this is done by a thiolase enzyme. So you're splitting the bond between the alpha and the beta carbon. Split that particular bond, and that is activated. This process is done by a thiolase. In this case, it's called acyl-CoA acetyltransferase. So this particular molecule is splitting, but it's also adding an acetyl-CoA to the rest of the molecule. Now, once the bond is split between the alpha and the beta, you are having this molecule. Okay, so this is a two carbon molecule, 1, 2, still bound to the acetyl-CoA as it was, but this acyl-CoA transferase is also adding another acetyl-CoA to the rest of the molecule. Now remember, we started at palmitic acid or palmitoyl-CoA, but you are removing these two carbons. So there were 16. So if you remove these two carbons, you're going to remain with 14 carbons, of course. So that is exactly what has happened here. So the thiolase has broken off this particular molecule, and you're left with another molecule still activated because it has a CoA enzyme CoA attached to it, but now it is a 14 carbon. And we know that a fatty acid that is 14 carbon is referred to as myristic acid. So myristic acid now becomes activated. Of course, it will be referred to as myristoyl-CoA. Myristoyl-CoA. This is just exactly what I've been talking about. So the first step here, you have an acyl-CoA that is activated. ADH, you're introducing the double bond, okay, between alpha and beta. The second step, you're introducing an OH, so the double bond goes, you're hydrating, of course, between the alpha and the beta. The third step, you are reintroducing a double bond at the beta. And the last step, you're breaking down fatty acids from plants, like the phytic acid, the phytic acid. Those are fatty acids from plants. If you are to ingest them in your diet, then definitely alpha oxidation will occur in the body. Of course, it mirrors beta oxidation. It's not any different from beta oxidation. But for omega, we're saying rarely occurs, but it majorly occurs in individuals who have issues with the pathway, or rather, yes, deficiencies in enzymes of the beta oxidation pathway. And you've seen of quite a number of enzymes that are involved in the beta oxidation. Okay, so that's clear for you guys.